In the next 10 minutes or so, I'll be talking about a technology that was developed at UCF back in 2013, and then how this technology is progressing. But before I start, I would like to give you a kind of background that we are in a dire situation to develop tools to enhance food security and improve nutrition. And UN goal number 2 is aligned with the same focus. Now, having said that, what are the things that is limiting our ability to produce food crops in a sustainable manner? Use efficiency of agrochemicals is one thing, and then losses due to biotic and abiotic stresses. Monocropping, soil degradation, and water scarcity are the contributing factors. And then globalization of diseases like pandemic. We have many pandemic in agriculture too. I'm going to talk about one of those today. Now, improvement, how can we use nanotechnology to improve crop productivity and assure food security? We can improve the fertilizer and pesticides. You know that these are the two major components that protects your crop. Increasing their uptake, translocation, and penetration in plants. Then efficient delivery, so nanotechnology can reduce the loss, and then minimize contamination. We always need regulatory approval for any kind of new pesticide we are bringing to the market. So how do we take care of the environment and human health? So those are the things we need to take care of it. And nanotechnology can be also used to monitor plant needs for smart fertilization. Now, if you look at human system versus plant system, we don't see much difference though. Plant has major avenues of taking things from outside to inside the plant, like root system that takes nutrient and water from the soil. Then the foliar means leaf, stem, tissues, they are exposed to the environment where we can apply agrochemicals, including nanomaterials, where they can be also uptaken through the leaf and stomachs. So having said that, I would like to focus on a particular element in our periodic table, that is zinc. Zinc is a micronutrient for human health and for animal system, as well as for plants. It is an essential element for protein synthesis and growth regulation. And also, if zinc deficiency is there, then you will lose crops and the quality will be compromised. That brings to the next slide, where UCF Zinkicide Nanotechnology is unique of its kind because it is a systemic bactericide. Systemic means that irrespective of the method of application, whether you put in a soil or you spray on the leaf, it is going to reach every tissue. That is how it is designed. Now, what is the uniqueness of this Zinkicide nanotechnology that reaches every part of the plant tissue? Now, if you look at the size of Zinkicide, it is about 4 nanometer in diameter. I'm sure that many of you are familiar with nanoparticles and then the definition of nano. So it is about a protein-sized particles that has the ability to travel within the plant tissue. Now, we are targeting a huge problem using this particle in our state, that is Huanglongbing, or also known as HLB. Not only Huanglongbing, but we have had, still we are having issues with the other citrus diseases, that is citrus canke. By the way, citrus industry in Florida used to be a $9+ billion industry. Productivity back in 2005 and 2006 was about 250 to 260 million oranges, boxes of oranges. Now we are down to about 60. So you can imagine 2/3 productivity loss is significant for our state economy. So the major culprit of this disease is a bacteria that is within the plant tissue. Basically, it is in the sugar transport system. And how do they get there? The culprit is the insect. It's a 2 to 3 millimeter size tiny insect that feeds on the phloem tissue, which is kind of vascular tissue that transport the sugar after photosynthesis. And during feeding, the bacteria finds its way to go from their guts to all the way the phloem tissue. And once they land there, they find like a dream place for them to grow and proliferate. As they grow and proliferate, they clog the phloem transport system. As a result, you will see the root loss, and root loss means eventually you will lose the productivity, your quality is going to be compromised, and in the end, the plant is going to die. So that's the most serious thing and serious consequences of this HLB. Now, why do we care about small particle technology, such as the one that I mentioned to you, Zinkicide? If you look at this slide, it gives you a kind of perspective. Why do we need this small particle? Well, if the particle size is about the 5 nanometer or less of protein size, it has the ability to move every parts of the plant tissue. A bigger size will not make it through to every parts of the plant tissue. Having said that, our particles, as you can see in this transmission electron microscopy images, is about 4 nanometer in size, less than 5 nanometer. Ideally, it has to be less than 5.4 nanometer. And in collaboration with a local company, Trademark Nitrogen, we are able to prepare agriculture grade, fully stabilized Zinkicide, which is available for research purposes. And if you look at the kind of cartoon or model of this Zinkicide, that it is designed to be completely degraded to zinc ions, which is the essential micronutrient. Thanks to our collaborator in Oak Ridge National Lab, they have done some fantastic job to understand why this size of particles is unique compared to others. Well, as you make smaller and smaller size particles, you create more surface defects. In quantum dot nanotechnology, for bioimaging and drug delivery and so on applications, you try to passivate the surface to make it perfect so that the optical and optoelectronic properties retained. In this application, you don't want that. You want just opposite. You want to create defects. So defects in our zinc oxide nanoparticle is the oxygen vacancy, and that vacancy serves as a catalyst to even break down water molecules to produce reactive oxygen species. And that is the unique properties of Zinkicide. Now, my colleague, Dr. Titar in Physics and Nanocenter, she did a fantastic job in demonstrating that how this Zinkicide zinc can move when we apply in the soil. Now, what I'm showing you is the EDS mapping, where, and the cross-section of the plant tissue, where the blue dots represent the metal zinc. So if you have different kind of sections of this plant tissue, you see control leaf versus upper leaf and lower leaf, and you see that it is distributed throughout the plant tissue, whether it is the woody part of it or the xylem or the phloem, it is everywhere. And naturally, the lower leaf close to the root system will have more population of zinc. So having said that, moving to the next slide. My colleague at Auburn University, who was one of our collaborators in the USDA grant, did a fantastic job comparing how this tiny small size zinc oxide particle compared with the CR41, which is traditionally used for rubber industry and so on, bulk zinc oxide. And we found that our Zinkicide is efficient in degrading the bacteria through multiple mechanisms. So it basically, the reactive oxygen species and the zinc, antimicrobial zinc, both plays an important role. And it takes part in lipid peroxidation, releases DNA, and so on. But he also did a very beautiful experimental in vitro studies where we had a microfluidic channel. You see that when you have bacteria population growing here in the upper channel with the untreated control versus bulk zinc oxide and Zinkicide, the red represents all dead bacteria and green represents live bacteria. You can see that the Zinkicide treated channel was completely red, no sign of green, meaning that it is very potent antimicrobial. Well, many things works in the lab and greenhouse perhaps, but not in the field. So transitioning or translating the technology that you start from a test tube in a chemistry lab all the way to the field trial involving industry is something important for us and in particular our students to understand. So we did trial in the Indian River area with on grapefruit, young grapefruit and Valencia on the ridge, 20+ years old. All of those trees had heavily affected by the HLB. Now, what I'm going to show you here is the yield response. Why do we care about yield? Well, growers who are producing these crops, they care about the yield and they care about the bricks, the quality. So if you look at the 2017 field trial data in a commercial grove, we see that as you increase the dose of Zinkicide, foliar application, as well as the soil application and combination of both, you see that is a dose-dependent yield response. So yield response measured as a harvested fruit weight pounds fruit per tree. And we also seen the 2017 and then 2018. It takes about 1 year to conduct the trial, every month application. And then also we did the pounds per tree when the full spray and drench was done and Valencia. So you see that spray plus soil applications works the best in most cases. So having said that, the fruit size also matters, because in particular grapefruit is the export quality grapefruit we produce in Florida, and our Florida grapefruits you can find many places outside USA. And Valencia also we did the acid bricks acid ratio, meaning the quality of the juice. We found that that the quality of the juice in fact improved and didn't compromise as you increase the application rate. And not only that, the fruit size also improved with our treatment in the dose dependent manner. Having said that, what is the future lying ahead of us? Well, I just gave you a glimpse of what nanotechnology can do. There are plenty of opportunities for us, material scientists, chemists, and the interdisciplinary team that I just showed you, that we work together at our MISA Center at UCF, that there are plenty of opportunities for us to think about how to design nanomaterials for future crop productivity and contributing to food security. But there are many roadblocks ahead: cost, food safety, environmental safety, health safety, regulation, and diversity of crops. So having said that, yesterday there was a press release, and I thought that many of my engineering colleagues will be excited about learning this opportunity, and there will be many more coming in coming years. So if you're interested, that NSF and USD NIFA invest 220 million artificial intelligence research initiative, and then USDA investing means they're excited about this technology. So having said that, I would like to acknowledge all of my colleagues. It's a multi-PI, multi-university initiatives who contributed, many students, postdocs contributed to develop Zinkicide. And I hope that I give you some idea about the Zinkicide. But if you would like to learn more about how does it work, does it kill? Yes, we have the evidence to show you that it indeed kills bacteria inside the plant. With that, thank you.